Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about carbohydrates. Carbohydrates are organic macromolecules that contain carbon, hydrogen, and oxygen, usually in a pretty predictable ratio of 1 carbon to 2 hydrogens to 1 oxygen. So the monomers that we are going to be using to make our carbohydrate polymers are called monosaccharides. Monosaccharides, of course, are joined together by dehydration synthesis to build covalent bonds joining them together, and that is how we are going to get the polymeric polysaccharides. You can see some prominent examples of important monosaccharides in the human body here: glucose, fructose, galactose, and then obviously the deoxyribose and ribose that are used as components of DNA and RNA molecules. Monosaccharides tend to contain somewhere between 3 and 7 carbon atoms, although most of them are going to contain either 5 or 6, usually in an adjoining ring pattern, as you can see here. You can see that some of these have more of a hexagon shape and some of them have more of a pentagon shape. The terms hexoses and pentoses are used in these regards, although you should be aware a hexose does not always look like a hexagon, and a pentose does not always look like a pentagon. A hexose is a monosaccharide that contains 6 carbons, and a pentose is one that contains 5. So another important thing that we have seen in a previous video is that monosaccharides are covered in hydroxyl functional groups, which are very polar, and that is going to make monosaccharides very polar themselves, and they will be very soluble in water because of this. So when we are going to start building polysaccharides by joining monosaccharides together, the type of chemical bond we are going to form by dehydration synthesis is called a glycosidic bond. In this regard, you can see 2 monosaccharides that are very nearby each other, and of course we can see the element of water that is going to be lost between them. So after dehydration synthesis occurs, we can see the glycosidic bond that is being formed here. We're not going to get into it too heavy, but glycosidic bonds can be formed in a number of different configurations and molecular geometries, which can make digesting carbohydrates a little bit complicated because generally you need a different kind of enzyme for each kind of glycosidic bond. But we're not going to worry about that outside of one example, which we will touch on later. So after you have joined 2 monosaccharides together, you get what is called a disaccharide. You can see what used to be 2 separate monosaccharides that are now joined together by a glycosidic bond. And when you're comparing the 3 types of disaccharides that are illustrated here, sucrose, which is a disaccharide of glucose and fructose, that's table sugar, sucrose here, lactose, which is a disaccharide of glucose and galactose, and then maltose, which is just 2 glucoses joined together. You can see that the way in which the glycosidic bond is illustrated here is a little bit different, and that's because they are different, again, in terms of how those bonds are configured in 3-dimensional space. So disaccharides are going to differ on the identity of the monosaccharides that are used to build them. And in the human body, there's a very limited number of very important disaccharides. The 3 here that are listed here are arguably the most important. So when we start building polysaccharides, we're going to be talking about carbohydrates that contain anywhere from just a handful of monosaccharides to several thousand monosaccharides joined together. We use the same umbrella term polysaccharide to describe these. There are 3 particular polysaccharides that are going to be especially important in the human body, so we'll go ahead and talk about those now. The first is called glycogen. Glycogen is a long-branched polymer that is entirely made of glucose, and it tends to be stored in skeletal muscle and in the liver. So the idea here is that as a storage molecule, when energy is abundant in the body, such as after you have had a big meal, excess glucose that is found in the bloodstream is instructed to be stored as glycogen, both in muscle and in liver. But when energy is limiting, such as when it's been a long time since you've eaten, the body will start breaking down this stored glycogen so that it can get access to glucose and bring the blood glucose back up. The way in which this process is regulated by hormones will be addressed in a much later video. Starch is another long-branched polymer of glucose, except this is the glucose storage method that plants use rather than glycogen, which is what animals use. So, as humans, when we eat starch-heavy foods like, for example, potatoes, these polysaccharides are going to be broken down by digestive enzymes that we find in the GI tract into individual glucose molecules, which can then be absorbed into the bloodstream and delivered to respiring cells throughout the body. Finally, cellulose, which is a structural polysaccharide that is a major component of plant cell walls. This is actually another polymer of glucose, but you can see here that the glycosidic linkages linking these glucose molecules together is very, very different. So this brings up kind of an issue for us eating cellulose in our diet. It's because this glycosidic bonds configuration is so different that makes us as humans unable to digest cellulose. It's because we do not have an enzyme that is capable of breaking these bonds. So cellulose is sometimes called dietary fiber or roughage because it goes all the way through our digestive tract without ever being digested, although there are, of course, beneficial health effects to ingesting a lot of dietary fiber. But the important thing here is that when we eat cellulose, we are not able to extract the energy out of it because we cannot break those glycosidic bonds, and we therefore cannot absorb any of this contained glucose into our bloodstream. Some animals can, especially animals that feed off of grass like cows and horses and things of that nature. Okay, so the most important function of carbohydrates out of some niche functions like cell recognition, of course, carbohydrates can be found in DNA and RNA molecules. The most important function of most carbohydrates in the body is that it is a source of energy for all of the cells in the body that need to make ATP energy. So in the diet, most of the carbs that we are going to eat and ingest are either going to be disaccharides like sucrose or lactose, or they will be polysaccharides like the starches that we eat. But the problem here is that these cannot be absorbed through the intestinal tract. So if we illustrate what this absorption is going to look like, we can see here that the lining of the intestines are made of a single thin layer of epithelial cells. You can see one of them here, and you can see another adjoining one over here. Up here you see the lumen of the intestine, which is just the hollow portion of the intestine through which digested food materials are going to be moving through. And then down here on the opposite side, you can see access to the blood supply. So if we have eaten a large polysaccharide, our goal here is going to be to absorb the ingested carbohydrates through this layer of cells and into the bloodstream. But like I said, this polysaccharide here cannot be absorbed as it is right now. So what's going to happen, a hydrolytic enzyme that is capable of breaking the glycosidic bonds that are adjoining these monosaccharides together is secreted into the lumen of the intestine, where it will break the glycosidic bonds of the carbohydrate into its constituent monosaccharide molecules. These can now be transported through the intestinal epithelia, first through the apical membrane so that the monosaccharide gets inside the intracellular fluid of this epithelial cell, and then through the bottom basolateral membrane so that it can then gain access to the bloodstream. Once in the bloodstream, these absorbed monosaccharides will be transported to all the body cells throughout the body that need the glucose or need the monosaccharide for energy. These cells will use that substrate as a substrate for cellular respiration, and they will use it in conjunction with oxygen that's also being delivered to make a lot of ATP that is necessary for cellular activities. Okay, that's going to do it for this video on carbohydrates. Here is a list of vocabulary terms you probably ought to be aware of. And then for checking your understanding, number 1, describe the chemical composition of a monosaccharide. Number 2, how are polysaccharides constructed? And number 3, what are some functions of carbohydrates aside from energy storage? You may want to do a little bit of research on your own in that regard. Okay, that's going to do it for this video. If you have any questions, go ahead and drop them in the comments section, and I will see you for the next video. So long.